Carbohydrates. Carbohydrates are macronutrients that can be found in a myriad of food, both healthy and unhealthy. Generally, we call them sugars. Most dietitians will recommend about 45 to 65% of your daily intake of calories to be consisted of carbs, or carbohydrates. But for today's video, we'll be focusing on the biochemical aspect of them. Chemically, as their name suggests, they're hydrates of carbon. Hydrate meaning water. In other words, they're made out of carbon and water in a ratio of about, for every 1 atom of carbon, 1 molecule of water, which gives us the following empirical formula. Now, as I said before, we can refer to them as sugars, but sugars, starches, and fibers are just the most common ways you can find carbohydrates in nature. And as you can notice, the three that I mentioned just now are vastly different, which brings me to my next point, classification. According to the degree of complexity, carbohydrates can generally be classified into 2 large categories, simple and complex. Starting with the simplest carbohydrates, monosaccharides. Mono means 1. And saccharides, essentially sugars. That's why monosaccharides can generally be referred to as simple sugars. They're the most basic form. You'll find carbohydrates as they're non-hydrolyzable. They're the monomers, the building blocks that make up more complex molecules. Examples of monosaccharides can be the most famous glucose, fructose, galactose, mannose, ribose that makes up DNA, etc. Oligosaccharides. Polymers that are made out of the association of about 2 to 10 monosaccharides. They can include disaccharides that are made out of 2 monosaccharides, trisaccharides that are made out of 3 monomers, tetrasaccharides that are made out of 4 monomers, etc. Now, oligos as a prefix comes from the Greek word oligos, which means just a few or scanty. As you can tell, that's very vague, which is why it's up to interpretation what you define as an oligosaccharide. In a lot of references, you'll find disaccharides as a separate category, generally under the same umbrella as monosaccharides, classified as simple. But for this video's purposes, I'm gonna define oligosaccharides as polymers that contain between 2 and 9 monosaccharides. For example, lactose, that is made out of both glucose and galactose, is both a disaccharide and an oligosaccharide. Polysaccharides. They're polymers of a higher degree of complexity, made out of more than 10 monomers. And generally, we can find 2 subcategories of polysaccharides. Homopolysaccharides made out of 1 repeating monosaccharide, like starch or glycogen, and heteropolysaccharides that are made out of a bunch of different monosaccharides. For today's video, we'll be focusing on the very basic of carbohydrates, monosaccharides. Now, structurally, they're composed of about 3 to 7 carbons. You're not really going to find any longer than that in your organism. And as such, we can name them according to the number of carbon by adding O's to the end. So a sugar that contains 3 carbons is a triose, one that contains 4 carbons is a tetrose, and one that contains 5 carbons is a pentose, and so on and so forth. Each sugar has one of 2 specific carbonyl functions, either an aldehyde or a ketone. A quick reminder, carbonyl functions are functions that have a double bond between a carbon and an oxygen. A ketone and an aldehyde function being one of them. The easiest way to differentiate between an aldehyde and a ketone is that an aldehyde is generally at the beginning of the chain. Think of it as a cake topper, and the ketone function is generally sandwiched between 2 other chains. Think of it as the filling between layers of a cake. Now, sugars that contain an aldehyde function are called aldoses, and ones that contain a ketone function are called ketoses. We can name them again, taking into consideration both the number of carbons and the type of carbonyl function as following. So monosaccharides that contain 4 carbons and an aldehyde function is an aldotetrose, and this is a ketopentose. It contains 5 atoms of carbon, pento, and a ketone function. Now, for most depictions of open-chain monosaccharides, you'll find them in Fischer projections. The principle behind Fischer projections is quite simple. Vertical bonds point backwards into the plane, and horizontal bonds point towards you, the whole arms coming out to hug you analogy. Now, what's most important to note here about Fischer projections of monosaccharides specifically, while it depicts open chain linear forms of monosaccharides, the molecule is not flat. It simply allows us to see the orientation of different groups easier while still more or less being true to its spatial conformation. Speaking of the orientation of different groups, let's talk about stereoisomerism. Stereoisomers are isomers, meaning they're molecules with the same empirical formula or the same constitution of atoms and groups, but they're different in the spatial conformation of these groups. Now, before we continue in the video, I want to define a couple of terms that you're going to find used quite commonly in this video and a lot of other videos about stereochemistry. I want to know the difference between chiral centers and stereocenters. Chiral centers, by definition, have 4 different substituents. Stereocenters, by definition, are atoms which the exchange of 2 substituents results in a stereoisomer. Just by definition, we can tell that chiral centers are stereocenters. Because they have 4 different substituents, the exchange of 2 will inevitably result in a stereoisomer. But not all stereocenters have to be chiral centers. In other words, chiral centers are just a small group of the pool of stereocenters. And let me give you an example of 2 molecules. This molecule has a chiral center. It has 4 different substituents, a hydroxyl, a hydrogen, an ethyl, and a methyl group. If we exchange 2, we result in a stereoisomer. This is both a chiral center and a stereocenter. So for the second molecule, the atom that I pointed on is not chiral, but the exchange of 2 substituents will result in a stereoisomer. This is the cis and trans conformation. Now, let's go back to monosaccharides. For stereoisomers, you're generally gonna find 2 types of stereoisomers, either diastereomers or diastereoisomers or enantiomers. Diastereoisomers are 2 molecules are stereoisomers that are not mirror images of one another, nor are they superposable. The easiest way to differentiate if 2 monosaccharides are diastereomers is just see if they're different in the positioning of the groups in one or more chiral center. For example, galactose and mannose are diastereomers. They're different in the orientation of the hydroxyl group in both the 2nd and 4th carbon. Apomers are a special case of diastereoisomers. They are different in the conformation of one specific stereogenic carbon. Let's say, for example, glucose and mannose are different in the conformation of the 2nd carbon, and glucose and galactose are different in the conformation of the 4th carbon. Enantiomers. Enantiomers are stereoisomers that are mirror images of one another, but they're not superposable. For carbohydrates, there is a specific notation that describes enantiomers, the D and L series. Technically, a monosaccharide belongs to the D series if the hydroxyl group of the last stereogenic carbon is pointing to the right, and it belongs to the L series if the hydroxyl group of the last stereogenic carbon is pointing to the left. An easier way to say that is just that the hydroxyl group of the carbon before last is pointing to the right, it's of the D series, and if the hydroxyl group of the carbon before last is pointing to the left, it's of the L series. Now, the general consensus is that sugars belonging to the D and L series are enantiomers. For example, D-glucose and L-glucose are enantiomers. They're mirror images of one another, but they're not superposable no matter how much you rotate them or you turn them. Now, while open-chain form monosaccharides do exist in the organism, they are fairly rare. Monosaccharides generally prefer to cycle because the cyclic form is a lot more stable. That's something that you'll find that is quite common in nature. Everything really just seeks stability. The cyclic form is generally dominant with about 99% of monosaccharides existing in cyclic form and less than 1% in open chain, and it's generally just a transitional stage. We'll talk about that a little bit more later. The cyclization of monosaccharides happens with the creation of an intramolecular, that means in the same molecule, hemiacetal bond. The hemiacetal bond is created by the addition of a nucleophilic oxygen from one of the hydroxyl group in the molecule with the electrophilic carbon of the carbonyl group in that same molecule. That results in a cyclic form. Literally takes one of two shapes. The pyranose form from the pyrene cycle, which is a six-membered heterocycle with an oxygen, or a furanose form, which is a five-membered heterocycle with an oxygen. You're not really gonna find cycles smaller than that. For aldohexoses, like glucose, mannose, and galactose, they generally prefer to cycle as a pyranose. But for other sugars, like pentoses, you're almost always gonna find them in a furanose form. Fructose also prefers to cycle as a furanose. Now, instead of leaving it in this awful cyclic Fischer projection, the most common depictions of cyclic form monosaccharides are in Haworth projection. In Haworth projection, the pyranose takes the form of a hexagon. This is more of an elongated Haworth than a classic one, where the inferior half is depicted as a little thicker to insinuate that it's closer to us than the superior half. Same thing about the furanose form. Now that we've established that, let's talk about how to convert a Fischer projection to a Haworth projection. It's very simple, just follow a couple rules. Let's go for example, glucose. Now glucose is an aldohexose, meaning that it prefers to cycle as a pyranose. The first rule that you need to follow is that everything that is pointing to the right in a Fischer projection will be oriented downwards in a Haworth projection, and likewise everything that is oriented to the left in a Fischer projection will be pointing upwards in a Haworth projection. For glucose, for the second carbon, the hydroxyl is pointing to the right, meaning in Haworth will be pointing downwards. The hydroxyl group in the third carbon is pointing to the left, meaning that will be pointing upwards in Haworth projection, and likewise the hydroxyl group in the fourth carbon is pointing to the right, meaning that will be pointing downwards in Haworth projection. The second rule is for carbons that dictate the series D or L, the one before last or the last stereogenic carbon. If the sugar is of the D series, the bond between the last two carbons will be pointing upwards. If the sugar is of the L series, the bond between the last two carbons will be pointing downwards. For this example, because this is D-glucose, the bond will be pointing upwards. Now we finished everything, but you notice that I did not really talk about the first carbon. After the cyclization, we have a new stereocenter, which means we have two isomers. These isomers we call anomers. The hydroxyl group can be pointing upwards or downwards. If the hydroxyl group is pointing the same way as the last bond between the last two carbons, it's a beta anomer. If the hydroxyl group is pointing to the opposite direction to the bond between the last two carbons, it's of the alpha anomer. So it can result in either one of two anomers. The process of cyclization is very much reversible. The cycle can open, go to the chain form, and reclose again to form the other anomer, and go back and forth between those three forms until it reaches a delicate balance. For glucose specifically, the balance is about 36% alpha anomer, 64% of the beta anomer, with the chain form being trace. The beta anomer dominates essentially the equilibrium because it's the most stable of the anomers. Now this cannot be said for all the monosaccharides, this is specific for glucose.